Okay, this is me. I'm one presenter. I'm here to present my work on evaluation of Musa Accessions indigenous to Benin Republic for resistance to black Sigatoka disease. Why is it not moving? Introduction. Musa species are important food crop cultivated in over 128 countries globally. This crop is ranked fourth most important food crop, following after rice, wheat, and maize. Hence, this tells us is very important in global food security. However, this crop is highly challenged. The production crop is highly challenged by both biotic and abiotic factors. The leading disease factor challenging the production of banana is the fungal organism called Pseudosporidium fijensis. This causes disease called black Circulatory disease. This disease is known to be the worst and most damaging disease of banana. The pathogen penetrates the surface, thereby causing necrotic damage and ultimately reducing photosynthetic yield for this area and causing yield loss. Now, to help address this increasing issue of food shortage and food insecurity, mostly across Africa, where World Fungal Map has shown that in most countries in Africa, more than 35% of the population are in chronic hunger. To help address this situation, we need to see how to select for breeding and for propagation crops that are resilient in the environment that can be resistant to pathogens and be high yielding. So this work was aimed at identifying local Musa accessions which are resistant to this disease. Material method. This research was carried in Meserete, a remote town from Porto Novo, the capital city of Benin Republic. Method. Soil collection. A total of 72 Musa genotypes were used for this study. The study layout was the augmented research design, as you can see on the screen, and the study was laid out using the augmented research design, laid out in Meserete, Benin Republic. After planting, the field normal agronomic process was carried out within watering and manure application. We use organic manure. Now, morphological data was collected 9 months after planting. The data collected included the data on disease incidence and severity. On disease incidence, we took data on symptom appearance, 12% leaf death to 100% leaf death. Whereas on disease severity, we collected data on youngest leaf spotted, number of Sunday leaf, index of Sunday leaf, area under disease progress curve, and disease severity index. Now, after the data collection, we did our statistics, including the means standard deviation. We did the ANOVA, the cluster analysis, the correlation, and PCA. Then we did molecular studies, which included DNA assertion, etaphoresis, DNA sequencing, and sequence analysis. From the results, the results show that symptom appearance on the list of the assertion studies range from 10 days to 36 days. Sorry, from 10 days to 6 days, whereas for 12% death range from 7 days to 45 days across assertions. Now, this suggests that these assertions react differently to this pathogen. Hence, some of the assertions may be resistant to the pathogen, whereas some are susceptible. Equally, on this severity index, we saw that youngest leaf spotted range from 2 to 6. Number of Sunday leaf range from 3 to 14. Index of non-spotted leaf range from 12.5 to 60, whereas disease severity index range from 13.33 to 50.0. On same hand, the area under disease progress curve was shown to range from 58 to 339 across the genotype studies. Now, after the mean analysis and everything, we tried to do cluster analysis with the data generated from the field. The data analysis showed the cluster analysis showed the presence of 3 clusters. Cluster A had 20 highly resistant accessions. These accessions on this part of the cluster has 20 highly resistant accessions, whereas cluster B in red has 16 highly susceptible accessions. Cluster C has 36 intermediate resistant accessions. Okay, when we did the farm analysis to see the correlation between the genes and the morphological data, after we used 2 genes anyway, we studied the BS1 gene, which is known to encode resistance to blood circulatory disease, and as well try to equally see the effect of another gene, disease gene, which has equally been implicated to contribute to blood circulatory resistance in Musa. Now, from the finding, it was shown that 300 BP allele of this second gene had a similar correlation with index of non-spotted leaf, youngest leaf spotted, and number of sunny leaf, and varied inversely. Sorry, please. With the infection index, which is this severity index. Okay, we equally did sequence analysis for the multiple sequence alignment. We observed that most of the susceptible accessions has missing sequences in some portion. So the sequence is too large, so we took some fractions, some portion of the result to paste to show here. Now it showed that most these three here actually highly susceptible accessions, and then they have been shown to have some missing sequences, both in this and other portion of the alignment, which we did using mapped software in EBI database. Now, in discussion, high significant variation was observed in blood circulatory disease incidence and severity parameters. Now, this significant difference suggests that these accessions respond differently to the pathogen, which suggests possible difference in sensitivity. Now, to confirm this, the 3 analysis grouped these accessions into 3 main clusters, which is typical of Musa sensitivity profile to blood circulatory disease, which include resistance, intermediate resistance, and susceptible. Now, this has equally confirmed that some Benin Musa accessions are actually resistant to blood circulatory disease. Also, the third observation was that the DNA sequence-based dendrogram equally gave 3 main clusters, similar to the morphological cluster, morphological tree, which equally show that these threats are gene related, they are gene controlled. Also, the farmed result equally revealed that the 300 BP allele, because the unspecified disease gene showed 2 allele, the 300 BP and 400 BP, but the 300 BP allele was highly correlated with parameters that indicates resistance to blood circulatory disease, which included the youngest leaf spotted and the index of non-spotted leaf, and inversely correlated with this severity index. They're suggesting that this gene may be playing essential role in blood circulatory resistance. Also, from the multiple sequence element result, it was shown that some of the susceptible accessions have missing sequences, which implies that there must have been a deletion event over evolution time that have made these accessions more susceptible than others. In summary, this study has identified blood circulatory disease resistant genotype among Musa cultivars, and has equally provided genetic tool which can be further verified and exploited for breeding, for future breeding purposes. Also, these resistant accessions that are observed in this study can be recommended to farmers and to breeders for propagation to ensure food security in the region. In spite of the milestone achieved in this study, because before now it wasn't known that any accessions were actually resistant, it was not classified. Now, but there are still areas that we need to go into. For instance, there is need to repeat this trial with larger number of sample and maybe with more number of markers and primers, which will help us do GWAS and other analytical tools, applied more of the analytical tools, which we learned in this study, in this program, in the lesson, can get more results from this study. Also, we need to understand the impact of blood level on blood circulatory sensitivity, because in the field it was shown that most of the sessions that showed thicker green coloration of leaf showed reduced disease spread across the leaf. So I hope that with future opportunities, we can do more studies on this to be able to characterize the local sessions based on their bloody group and genomic group as well, and be able to ascertain the impact of bloody level on blood circulatory resistance. Acknowledgement. I want to acknowledge the Margaret Inter-African Research Group that funded this, the European Union, the African Union, my host institution, University of Abomey Calavi, Benin Republic, my home institution, Ebonyi State University, and other partner institutions. And I equally want to acknowledge the opportunity given to me by the genomic group to present this work. Thank you so much.